Thank you very much for the invitation and introduction, Epicha. Okay, so good morning, everybody. Today, I'm going to talk about International Consortium of Rice Insertional Mutagenesis, about resources, utilization, and beyond. In Taiwan, we generate about more than 100,000 tDNA tag lines, with about 50% of them already have phylogenetic sequence data, which is searchable, and also phenomics data also searchable. So today, as a member of International Consortium of Rice Insertional Mutagenesis, I'd like to tell you our progress. As you all know, that current status, as you know that rice mutant will be very interesting for rice functional study. So this is many of these data come from a recent book chapter. It's mutant resources for functional analysis of rice genome. So currently, there are 12 populations using tag vectors, including tDNA, TOS17, ACDS, SPN, and also 6 populations using chemical or physical mutagens, including fast neutron, gamma ray, ion beam, DEB, EMS, MAU, and sodium azide. Altogether, we have about 800,000 lines worldwide, all the collection, and variety used. I classify them into Japonica, Indica, so please pay attention that including chemical physical mutagens or tag vectors, people pay more attention to Japonica rice. So there are Nippon berry, Tainan 67, Dongjin, Huayang, Kitake, Yuki Hikari, and Kimazu. And in Indica, there are IR 64, Zhonghua 11, Zhonghua 15, and Kasala. So I will use 2 slides to introduce all the library names, which country, and genetic background, mutagen, and website. So I noticed that Epicha distribute all the slide abstracts to all of you. So I will later on I will give him all this information so that if you want to trace all these resources, because the website are here, it's easy for you to find out. So please notice that there are tDNA, TOS17, ACDS, so with website here, and for many countries, including countries in Asia where rice production are very important, as well as in other countries which also contribute to this important research. So as well as for the tagged population, also ACDS, SPN, altogether, there are 12 populations. Please notice that many people didn't pay attention to Dr. Ray Wu's work in Cornell University. He was using Nippon berry, and with ACDS, paper were published here, and after he passed away, we tried to find out all the seed collections, although there are phylogenetic sequence data published in this paper, but we cannot find out the seeds. And then starting from here, that's the 6 population by either physical or chemical mutagens, including efforts in Erie, in UC Davis, in NIAS, Zhejiang and Terry in Taiwan, and Oresa Base in Japan. So again, here I list all the genetic background, I mean the variety we use or mutagen used. And here are the websites which Epicha will give you all this information later. So because we think that phenomics data are very important, so right now I reorganize the table I just showed you. So here are tagged mutant population with phenomics data. So including TRIM, TOS17, RND, Oresa tagline, and this RDA from Korea, including tDNA or TOS17, or ACDS. Altogether, for those that with phenomics data, it's still less than 200,000. Then for the tagged mutant population without phenomic data, okay, as we indicate here, in fact, some of them already have a lot of lines ready to distribute. However, because many of them are GN rice, okay, so in many countries it is not too easy to grow them in big area, and rice breeders work in the field to grade the phenotype. So you may notice that, okay, we have only one half of them with phenomics data. And then for the non-tagged mutant population, 2 of them already with a searchable, okay, phenotype database. Okay, so altogether with these 6 population, we may notice that in Erie, 64, in Zhejiang University, okay, and also in NIAS in Japan, UC Davis, USA, in Taiwan, Terry, and also NIG in Japan, using all sorts of different mutagens. So for the tagged population, such as tDNA, TOS17, ACDS, and SPN, most people are now using sequencing of the integrated sites so that FST data may be searchable. And then for the physical deletion mutant, people are now using PCR of poor DNA or genome DNA array to try to find out the mutant which occur, okay, in the candidate gene region. As well as for the chemical point mutagens, then people are using either TLIN or NGS coupled with multiple dimension pooling, DNA pooling. So we used 3 years ago, we were using at that time of the available TOS17 database and tDNA insertion database and ACDS. So we may notice that in in a 1 bag sequence, so here is about 120K in chromosome 9. Okay, so please notice that there are many TOS17 integration sites here. The triangle position meaning either forward or reverse of TOS17. So this means that, okay, TOS17 have a very hard integration site, but then for this color, okay, this is tDNA or ACDS. It's basically evenly distributed in this area. And then we also analyze chromosome 9, okay, whereas chromosome 9, we divide that into 20 slot, okay, regions. So here are the non-TE genes density in that slot or TE density. So here is the centromere. So please notice that for TOS17, we highlight those that's less than 1% integration site located in that slot. Once again, 1 out of 20. So 3 slot is less than 1%. But however, toward the end, as you may notice here, 2 slots, so it's 10% out of the whole chromosome 9. There are more than 50% of integration in chromosome 9 located in this area, where the non-TE gene is the highest. But for compared with tDNA, please notice that there are even integration in the centromere region or in the nearby region where TE density is still very high. So there's only 1 slot which is low. So we see tDNA integration in heterochromatin region, and also different tagging vectors should be used to generate mutant. As I just mentioned, altogether, international effort, people were using tDNA, ACDS, TOS17, and SPN. So altogether, with so many different tags, we hope that we are able to saturate the rice genome. In the previous log, okay, Chifa was using 99%. So although we are using the same equation, but the number are different. So with the genome size of rice and also rice gene density, if we want to, if possible, to saturate 95%, here's the data. Okay, so 460,000. Sorry. Okay, for 400,000. Okay, so altogether, okay, for the 12 rice chromosomes, here are the number of genes in each chromosome, and altogether the number is here. And here are the insertions, okay, of DS insertion, SPN, tDNA, and TOS17. So altogether, the number is right here. So very similar to what Chifa just mentioned. Out of the number that we calculate out right now, okay, although we have so many efforts international-wide, but in terms of flanking sequence data, searchable data, right now it's only 1/2 of the estimate number. If we compare with adoptive genome, okay, using the same equation, here is the number. But however, for this worldwide adoptive forward and reverse genetics resource links, there are more than 30 links here, and I just write down 4 of them. It's already almost saturated. So this means that right now for adoptive people, for most of any single candidate gene, you may find out 2 to 5 on average of the tDNA insertion in that gene region. But right now for rice, we are still far away from that point. So right now I'm going to use some example, including forward and reverse genetics way, okay, to study some genes. So although there are a lot of them, about 100 papers already published, I will only use 1 TOS17, 1 DS, and 1 tDNA research work. This is work by Niels. Okay, so TOS17 is published in 2001. It's still in the very early stage of IRGSP sequencing work. So they are working on this rice viviparous mutant. So it's germinate while still in plant. So because with the flanking sequence data, okay, so they are able to demonstrate right away that the TOS17 is integrated in a gene responsible for ABA synthesis. And also for that specific mutant, there's quick water loss and fails to accumulate ABA under drought. So that's the first example. The second one, okay, is using DAs published in 2003, okay, from CSIRO. So they are using tDNA tagged mutant line to try to find out genes responsible for branched florregulars. So here is the wild type, the panicle, and this is the mutant. Okay, it's just like a rod. So Nara, okay, try to open this. Okay, so that's the photo. So if we zoom in and use SEM to take a look at the flowered, so here are the wild type, but here are the floret mutant. So you may notice that there is no floret, i.e. spikelet at all. So at that time, 2003, so there are already PAC sequence data available. So they are able to demonstrate that the DAs were integrated in this area, which is a gene, okay, contain the AP2 domain, which is, so they suggest that that is an analog of the maize transcription factor gene branched circulase, BD1. And then I will use tDNA, okay, as example. This is published in 2008 after the finish of IRGSP. In Taiwan, I just mentioned we generate almost 100,000 tDNA line. Some of them have only knockout function, but most of them also have activation tagging function. So out of this, here are 4 mutant line, okay, and all of them have activation in GA2 oxidase in different genes, but you may notice that some of the mutants are severe drop. Some of them are still okay. So we then studied, so we found out that there are totally 10 GA2 oxidase genes in rice genome. So totally 10. So we're using tiller development stage or early germination stage. And so we are very sure that these 10 GA2 oxidase genes express in differently. So then cooperate with Dr. Seward in the States. We also measure, the mutant line, the GA1 and GA197. We then found out that there are, in fact, two clusters of GA2 oxidase, including C19 or C20. The C19-1 are larger, and for those that mutation occur in C20 cause less severe GA deficient phenotype, and they may be useful for the second green revolution, because for those mutant lines, they have much more tillers. They are a little bit shorter, drop, not severe drop, only semi-drop, etc. So I'm still using TRIM database. As I just mentioned, we have phenomics data which are searchable. And tomorrow I'm going to give another talk, how to use this phenotype, how to use this not just phenotype, including flanking sequence and everything. So you can notice right away this is a recessive trait, because for all of them we plant 3 by 4, so 12 T1 plants in the GM field. And this one, apparently that is a dominant trait. But for these two, we have only shown one mutant type. But in fact, many of them, they show 2 or 3 out of these 12 plants. So they are wild type and type 1 mutant, type 2 mutant, or type 3 mutant. So we can see that for those that with only one type, it's about 16%. With 2, about 2%. With 3, we still have even up to 4. So this means that mutation must include, those induced by integrated tDNA, the movement of TOL17. By the way, the material we are using is Taiwan 67. They almost will not jump. TOL17 almost will not jump in the material we used, as well as other sequence changes of unknown origin. Right now, worldwide, we already found out that the tagging efficiency, i.e. the co-segregation of phenotype as well as tDNA insertion, tagged flanking sequence, it's very low. It's around 5% compared with about 1/3 in the abducted tagged population. So we know that there must be high somoclonal variation in the tagged rice mutant population. So right now I will introduce some of the recent work using NGS to try to find out what are the somoclonal variation. First, this is a work by group of TOL17 group, which published early this year in PCP. So they sequenced 3 different TOL17 lines, and they found out many SNPs and very small indels. And here are the rates that they found out. Quite low, but so here are the 3 lines I just mentioned across the 12 rice chromosome, where you may see that for each specific TOL17 line, where are the integration sites. And they're also using PCR to confirm the sequence changes. Another paper is by the French group. They also use TOL17 lines. Also this means also Nippon berry variety. So they also use NGS. So they found out there are 34 new insertions. So here are the list of the new insertion. And here are all these 12 chromosomes. Where are the insertions are? So of course, as you may know, this is Nippon berry. So the abundant LTR retrotransposon will be top 17. There are totally 11 of them, but they are also Rari 2, Rari 3, and also some of them are mites. Also, they also use PCR, okay, to confirm their result. And also, I'd like to mention some of our work in the TRIM mutant in Taiwan. So we were using tealing of a gene region. So the estimated mutation rate, okay, is very similar in the same log as the one I just mentioned, okay, published by Japanese group, top 17 group, in the same log range, and there are many small in there, around 20 base pair in there. But the longest deletion we found out is 101 kb. It's very long. It's about 20 genes were deleted. And so that specific mutant is seeding lethal, so they may grow up to about 3 cm, and then they are lethal within about 2 weeks. And also one of the mutant is tetraploidy. I went through our TRIM GM field with a senior rice breeder, so he pointed to one mutant and told me that I think it might be polyploidy. In fact, I don't believe on him at that time, but I was very polite. I said, okay, I will bring one tiller back to my institute. So the next day we use flow cytometer run through. Bingo, that is really a tetraploid. So for the rice functional people, I hope we all will very much appreciate the effort by rice breeders. So here are we also perform NGS of several of our mutant lines. Here are one of them, so that although we have Franklin sequence data, but the tDNA integration do not co-segregate. Okay, this is dwarf, short panicle, small seeds, and many tillers. So we found out deletion, we found out insertion, we found out SNPs, as well as like this one. We can easily demonstrate this is a find, very similar to what Hirochika's group and Olivier Pernal's group mentioned. Okay, I just mentioned 2 papers. Okay, so because we have very high molecular variation and we have very low tagging efficiency, so I think for rice community, we really have to develop novel method to generate transgenic rice plants. So first, that transformation method have to without callus formation, so that they will not went through the proliferate rice callus growth, so that the molecular variation will be decreased. So such as maybe we will suggest developing panicle, like years ago, Laplace's people, they were using root, okay, to generate transgenic plant, but later on they are able to using flower deeping method, and also maybe development of new carriers such as nanoparticle will be another novel method, so that we may generate more transgenic plant, okay, rice plant, but very low molecular variation. Thank you very much.
Thanks, Caroline. We have about 5 minutes for questions. Don't forget to fill in. One question there. Please introduce yourself. The microphone.
Thanks for your presentation. I have a question about the calculation to saturate rice gene index the mutant population. So you use the gene from the whole annotation from the rice genome annotation project. The gene number is more than 50,000, but the gene has the expression evidence such as the EST and POLAC cDNA, maybe the number is reduced to 32,000. And then your calculation will be changed. And also tDNA has preference to insert in the U chromatin with the active gene expression. So then maybe you have to change the number of the expression to saturate the nice tDNA insertion of mutant.
Yes, I agree with you. So, I mean, this is just a calculation, but as I just mentioned for abductis community, okay, although the calculation number, okay, is here, but they generate much more than that. Okay, so this is our, I mean, for us, okay, we have to have as many as possible, but right now we have much less than what we expected. So I can only mention this. I'm sorry, here in the platform, we cannot really hear very...
The reason is like, of the estimated gene, around at least 10,000 is not real gene, the pseudo gene. So do you have include the pseudo gene for the functional analysis or not? That's what I have a question about your estimation.
Caroline, Kifa-san, so you also mentioned about insertional mutagenesis. And I think both of you would have the same problem of transferring material if you're going to have the international collaboration on these kind of things. So would you think of the killing population that we have been generating so far, would that be a better venue for collaboration? Killing population, radiation by fast neutron?
Yes, I agree with Epichar's point. Okay, so as I just mentioned, okay, in fact, we have tagged or non-tagged population, and we may use several methods, including tilling here. And yeah, Epichar is talking about this. But as I just mentioned, okay, right now, okay, so you see Davis, they already have this service. But yes, I also agree with you that if we, okay, the whole rice community, if we also develop, okay, all the bulk up most of these available mutant, and then using tilling method, so that's fast, and then try to find out the candidate mutant that we are interested in. Yes, I agree with you.
Thank you. Thank you. If there's no questions for that, I would like to thank Caroline and give her a big hand, please.
Thank you.